Hello everyone, welcome to the Swayam course of Genetic Engineering and its recent advancement. I am Dr. Richa Sahu, working as an assistant professor at MIT Institute of Biotechnology, MIT University, Chhattisgarh. Today we will be talking about mammalian artificial chromosomes, also known as MACs. So till the previous lectures you have talked about I think two different artificial chromosomes, YAC and BAC, bacterial artificial chromosomes and yeast artificial chromosomes. So today we will be talking about mammalian artificial chromosomes, but before starting MAC, let's brush up a little about artificial chromosomes. So basically artificial chromosomes are the synthetic genetic constructs designed to replicate and function like natural chromosomes within a cell. They are primarily used in genetic and biotechnology research to study various aspects of genetics, gene regulation, gene therapy and other therapeutic applications. There are several types of artificial chromosomes, each with its own specific characteristics and application. Talking about the types of artificial chromosomes, the first type is bacterial artificial chromosomes or BACs. BACs are artificial chromosomes that replicate in bacterial cells such as Escherichia coli. They are typically used to clone and study large DNA fragments, making them useful for genomic mapping and sequencing projects. These are one of the earliest artificial chromosome constructs that were made. Next type is yeast artificial chromosome or YACs. Yeast artificial chromosomes are artificial chromosomes that replicate in yeast cells as the name suggests, like Saccharomyces cerevisiae. Yeast artificial chromosomes can carry very large DNA fragments or inserts, making them very valuable for the cloning of large genomic regions and for studying eukaryotic gene regulation. YACs have been extensively studied for the eukaryotic genetic systems. Next is human artificial chromosomes or mammalian artificial chromosomes. MACs and HACs are same. Human artificial chromosome or mammalian artificial chromosomes are designed to mimic natural human chromosomes. They are used for various purposes including gene therapy and the study of chromosomal disorders. HACs can carry a very large stretches of human DNA and have the potential to deliver therapeutic genes to patients. They are majorly used for gene therapy. Next is plant artificial chromosome or PACs. PACs are similar to BACs and YACs but are used for plant genetics and biotechnology research. They are employed to clone and manipulate plant genes, study plant genome organization and develop genetically modified crops. Next is synthetic chromosomes. Some researchers are working on creating entirely synthetic chromosome from scratch rather than modifying the existing ones like PACs, YACs or BACs. These synthetic chromosomes may have unique features or functions designed for specific applications in biotechnology and synthetic biology. Next is mini chromosomes. Mini chromosomes are again artificial chromosomes which are smaller artificial chromosomes designed to carry specific gene or genetic elements. They are used for specific purposes including gene expression studies, protein production and the development of genetically modified organisms. Next in line are episome. Episomes are also a type of artificial chromosomes. These are the genetic elements that can exist either as a part of chromosome or as an independent plasmid. They can replicate automatically or autonomously and carry genes that provide selective advantage to the host organism. While not always considered as true artificial chromosomes, they can function in a similar manner and they can be easily used for similar applications. So artificial chromosomes play a very critical role in genetic research, biotechnology and gene therapy and other therapeutic application by allowing the scientist to manipulate and study genes and genetic elements more effectively as they mimic and replicate the human chromosome or the natural chromosomes. Their versatility and ability to carry large DNA fragment or constructs make them a very valuable tools in various fields of biology and genetic engineering. Starting with mammalian artificial chromosomes. So, mammalian artificial chromosomes are the synthetic chromosome-like structures designed and engineered for various purposes in molecular biology and genetic research. They are typically created by combining essential genetic elements to mimic the behavior of a natural human chromosomes within the mammalian cells. MACs have been developed to serve several purposes including gene therapy, functional genomics and biotechnology applications like treatment of various genetic disorders. Talking about the structures of mammalian artificial chromosome, as I talked earlier, they are designed to mimic the natural chromosomes, therefore, to resemble as a natural chromosome as closely as possible, they typically consist of a centromere, which ensures proper segregation during cell division, and telomeres which protect the end of the mammalian artificial chromosomes. Additionally, they contain origin of replication sequence to enable their replication within the host cell. So, they have a centromere, they have a telomere and they have an origin of replication sequence to mimic perfectly as closely as the natural chromosomes. What are the genes and genetic elements that are present there or the MACs can carry? MACs can carry large DNA fragments, gene of interest or the entire gene clusters. Researchers can have inserted or researchers can insert specific genes or regulatory elements or regulatory genes or other genetic sequences into mammalian artificial chromosomes to study the function or to express therapeutic genes to study in gene therapy applications. Mammalian artificial chromosomes have been a topic of scientific research and development for several decades. Various research to develop artificial chromosomes dates back to I think early 1990s. The artificial chromosomes are engineered to mimic the structure and function of natural chromosome found in mammals. They are often used as a tool for various biotechnology applications including gene therapy and genetic engineering as mentioned before. So, let us talk about the history of mammalian artificial chromosome, how the research started, what were the type of the mammalian artificial chromosome they were first synthesized. So, talking about history, it started with in early 1990s. The concept of creating artificial chromosomes in mammals emerged in early 1990s. Researchers began to explore the idea of designing synthetic chromosomes that could carry specific gene of interest and replicate within mammal cells. Not only that, they should mimic as a natural chromosome inside the host mammalian cells and which can be later used for various therapeutic and genetic engineering based applications. In 1997, in this field, the first synthetic mammalian artificial chromosome was synthesized or created, known as human artificial chromosome or HAC. This achievement was made by Hoshi and his colleagues in Japan. They successfully developed a small functional artificial chromosome capable of carrying genes and replicating inside the human cells. They had centromere, telomere and original replication in the artificial chromosome. Then in early 2000, the development of HACs continued to progress with researchers improving their stability, efficacy and capacity to carry larger gene fragments. These artificial chromosomes showed promises of gene therapy and other application as they could potentially carry therapeutic genes and introduce them in human cells without disrupting the natural genome. Talking about mid 2000s, researchers in various countries including United States of America, Japan, United Kingdom and other European countries made significant advancement in the designing and construction of such human or mammalian artificial chromosomes. These developments included the creation of modified versions of HACs and other synthetic chromosomes, as well as exploration of different strategies to introduce them into mammalian cells. So, in the mid 2000, most of the research was focused on modifying or improving the older version of HAC so that it can be more efficiently used in the mammalian cells without interfering with the natural genome. In 2010, around 2010, the use of artificial chromosome expanded beyond the gene therapy to other applications such as bioproduction of pharmaceuticals and the study of gene regulation and chromosome biology. In this era, researchers explored the use of synthetic chromosomes or artificial chromosome in model organisms other than humans, including mice and pigs, so that these artificial chromosome can not only be used for gene therapy or in therapeutics, rather than that it can also be used for the production of important biochemicals or bioproducts or pharmaceutics or in the host cells like mice and pigs. Talking about recent years, the data that is available or quoted by me here in this lecture is still early 2022. In early 2022, research in the field of mammalian artificial chromosome continued to advance. Techniques for constructing and manipulating these artificial chromosomes became more sophisticated, and their potential application in biotechnology and medicine continued to expand. It's important to note that the development of mammalian artificial chromosome or MACs or HACs is an ongoing and evolving field of research. Till now, a lot of research has been done which might not be quoted in this lecture. These artificial chromosomes hold great promise for the wide range of application, but they also come up with challenges like stability, efficiency, safety and ethical issues when used in therapeutic context. So, talking about construction of MACs or mammalian artificial chromosomes, there are various methods that are quoted for the construction of mammalian artificial chromosomes, and there are various key steps that are involved in most of the construction methods. So, let us discuss the the key steps that are involved, or the key steps or techniques that are involved for the construction of human artificial chromosomes or mammalian artificial chromosome. So, the first step is the selection of centromere in the construction of MACs. For the selection of centromere, centromere is a very crucial event of the artificial chromosome or any chromosome, because it ensures proper segregation during cell division. Choosing an appropriate centromere is a fundamental step. Many HACs use the alpha satellite DNA as a centromere region. After the selection of centromere, the next step is selection of telomeric sequences. As we know, telomeres are very important for the protection of the ends of the chromosomes. Telomeres are the protective caps at the ends of the natural chromosomes. Telomeric sequences are essential for the stability and replication. Synthetic telomeric sequences are often incorporated into mammalian artificial chromosomes. After the centromere has been selected, telomere sequence has been selected, the next step is the construction of mammalian artificial or human artificial chromosome vector. The human artificial chromosome vector is the backbone on which the synthetic chromosome will be built. It usually contains the telomeric, the centromeric and other necessary sequences. Different methods can be used to construct this vector, such as yeast artificial chromosome or YACs, bacterial artificial chromosome BACs or plasmid based vectors. After the HAC vector has been constructed, the next step is the introduction of the genetic material. Once the vector is prepared, the desired genetic material such as specific genes or regulatory elements can be introduced. This can be done through various techniques, including restriction enzyme based cloning, PCR based method or gene synthesis. After that, the next step is cell line preparation. To create a stable human artificial chromosome carrying cell line, the modified HAC vector is typically introduced into a target mammalian cell line. Various methods can be used for that, like transfection, electroporation, viral transduction or liposuction. After the cell line has been prepared, the next step is selection and screening. Cells that successfully integrate the HAC construct are selected and screened to identify those containing the artificial chromosomes. This can involve using selectable markers or fluorescent tags on the HACs. So, you can use fluorescent tags or any selectable marker to select the cells which have successfully integrated the HAC vectors inside them. The next step is verification and characterization. The newly established cell line with the HAC should be thoroughly characterized to ensure that the artificial chromosomes is stable, functional and properly segregates during the cell division. Techniques like fluorescence in situ hybridization, FISH or karyotyping can be used for the verification in this step. After the verification and characterization has been done, the next step is amplification and maintenance. Once the cell lines with HAC vector have been established, the cell line containing the HACs can be expanded and maintained under selective conditions to ensure the persistence of the artificial chromosomes. They have to be properly maintained in a good culture condition so that they are stable in nature. Next is the functional assays. Depending on the intended application for which this HAC have been established, functional assays can be performed to assess the functionality of the gene that is carried by the HAC. It is just to check whether the genes that are carried by the artificial chromosome or the mammalian artificial chromosomes which are inside the cell lines are working or not. So, these were the key steps which are used for the construction of mammalian artificial chromosomes or human artificial chromosomes. Talking about the methods of constructions of MACs or HACs, constructing a human artificial chromosomes or MAC is complex and evolving field of genetic engineering, and there are several methods used to create them. Here are some key methods that are used or employed for the construction of HAC artificial chromosomes. First is the top down approach. Bacterial artificial chromosomes modification is done in top down approach. Here BAC, which is a circular piece of DNA that can be propagated in bacteria, are often used as a starting point. Specific genes or genetic elements are inserted into a BAC using molecular cloning technique. These modified BAC can be then transformed into human cells to potentially convert them or to potentially form a human artificial chromosome. The next method that are used is bottom up approach. It is a de novo synthesis of human artificial chromosome. In this approach, synthetic DNA is designed and assembled in vitro to form a functional chromosome. This synthetic DNA can be based on natural DNA sequence or entirely novel sequences. This method allows for precise control over the genetic content of the human artificial chromosome. The third method which is used is Cre-Loxp recombination. The Cre-Loxp system is used to insert specific gene or genetic elements into the existing chromosomes, including HACs. Loxp sites are introduced into the chromosome, and the Cre enzyme is used to catalyze recombination events, thereby integrating desired genetic material into the human artificial chromosomes that are constructed. The next method is using yeast artificial chromosome. In this method, yeast artificial chromosomes, which is another type of artificial chromosome, are used as a basis for the construction of human artificial chromosomes. Genetic elements can be inserted into yeast artificial chromosomes, and these modified yeast artificial chromosomes can then be introduced into human cells to potentially form HACs. Next method is centromeric engineering. A crucial component of HAC, as mentioned earlier, is a centromere, which is essential for proper chromosome segregation during the cell division. Various approaches have been developed to engineer functional centromere for hacks, including the incorporation of a specific DNA sequence or protein that mimic natural centromeres. Next method is episomal system or usage of the episomal system. Some human artificial chromosomes are designed to exist as episomes, which are genetic elements that can replicate independently inside the host genome. These episomal human artificial chromosomes are maintained as extra chromosomal elements in the cell. Next method is telomere addition. Telomeres are the protective cap at the end of chromosome. Adding telomere sequence to artificial chromosomes helps maintaining their stability and integrity. Telomere sequence can be added using molecular biology technique. Finally, functional testing. After construction of potential human artificial chromosome, as mentioned in the steps as well, functional testing is a very crucial step. This is to ensure that it behaves like a normal chromosome. This involves assessment or assessing its stability, replication, segregation and gene expression patterns. It is very important to note that the construction of hack is a very complex and technically challenging process, and the specific method which is to be chosen may depend on the intended application the researcher has selected and the expertise of the researchers which are involved in this work. Additionally, ethical and safety consideration must also be taken into account when working with synthetic chromosomes, specifically or especially when considering their potential use in gene therapy or biotechnology applications. So, why these complicated methods are used for the construction of such complex molecules like human artificial chromosome or mammalian artificial chromosome? Because they have a wide variety of application in the field of biotechnology, genetic engineering and therapeutics. Talking about the applications of human artificial chromosomes or mammalian artificial chromosome, the first and the very potential application is gene therapy. Mammalian artificial chromosome can be used as vectors for delivering therapeutic genes into the host cell for the treatment of various genetic disorders. They offer a more stable and controllable platform for the gene therapy in comparison to the traditional viral vectors. This is particularly valuable for treatment of the patients with genetic disorders which are life-threatening. The next application is functional genomics. MACs or mammalian artificial chromosome can carry a very large genomic fragments and entire genes, which allows researchers to study the function of a specific genes or regulatory elements in their natural genome context. This helps in understanding the gene function and regulation. So, functional genomics is basically the field of genetic engineering which deals with the functions of gene. They either have a gene, they don't know the function, or they know the function, they have to identify the gene. So, in this kind of application, when we are working with the genomic function or function of a specific gene or the regulation of the gene structures or studying the regulation of gene expression, such artificial chromosome or mammalian artificial chromosome are a very important tool. Next application is transgenic animals. MACs can be used to genetically engineer or to generate transgenic animals with precision. They are a very important tool for specific generation of transgenic animal which are very specific as per the need of the researcher. The researchers can introduce MAC containing specific gene of interest or gene clusters into the germline of the animals to study genetic function, gene or disease models for the biotechnological purposes. Example, producing human proteins in animal milk. This is a very important application with respect to the generation of transgenic animals. Next application is cell line engineering. Mammalian artificial chromosome can be used to engineer mammalian cell lines for bioproduction purposes. For instance, they can be used to create cell lines that stably expresses specific proteins or enzyme for pharmaceutical or biotechnology application. So, they can be very well used if the cell line can integrate hack vectors these cell lines will be engineered cell lines, and these engineered cell lines can very well be used for the synthesis of specific proteins or enzyme, which can be used for the field of pharmaceuticals and other biotechnological applications. Next is the stem cell research, a very important application. MAC or mammalian artificial chromosomes can be introduced into pluripotent stem cells, that is embryonic stem cell which can make all the 3 germ layers, to study developmental process, model diseases, or to generate cell lines for regenerative medicines. Again, there are ethical issues associated with this application which has to be considered, but it is a very important application. Next is the epigenetic studies. MAC can be designed to carry modification or epigenetic marks to study their effects on gene regulation. This is important for understanding how epigenetic changes impact of disease and development. Next application is gene expression studies. Researchers can use mammalian artificial chromosome to investigate the regulation of gene expression by introducing reporter genes or other regulatory elements into the chromosomes. This helps in understanding gene expression dynamics. Next very important application is drug discovery. Mammalian artificial chromosomes can be used in drug discovery and development by creating cellular models of disease. Researchers can introduce disease associated genes into cells using mammalian artificial chromosomes and screen for potential drug candidates. Next is biocontainment. In synthetic biology, MACs can be engineered with biocontainment mechanism to prevent spread of genetically modified organism into the environment. It is a very important application. Next is chromosome engineering. Mammalian artificial chromosome can be designed to study fundamental aspects of chromosomes biology, such as centromere and telomere function, replication and segregation. Next is gene regulation studies. Mammalian artificial chromosomes can be used to investigate the role of non-coding RNAs, enhancers and other regulatory elements in gene regulation. Next is artificial chromosome library. This can be used for the construction of artificial chromosome libraries. Researchers can create libraries of various types of mammalian artificial chromosomes with different genes and genomic regions, which can be used for systematic functional genomic studies, for therapeutic application or other gene regulation based studies. Stability of mammalian artificial chromosome is good. Its relative stability is good in comparison to the traditional plasmid based vectors. Plasmid can easily be lost or rearranged into the host cells, but MAC tend to remain more stable over time in the human genome. But there are certain challenges associated with human artificial or mammalian artificial chromosomes. While MACs offer numerous advantages, their creation and maintenance can be technically challenging and tedious. Ensuring proper segregation during the cell division, avoiding interference with the host cell function are important considerations. Apart from that, there are few ethical issues that also has to be dealt before working with mammalian artificial chromosome for therapeutic purposes. So, in conclusion, mammalian artificial chromosome represent a very powerful tool in genetics and molecular research, offering a means to manipulate and study mammalian genome with greater precision and stability. Their development has a potential to advance field such as gene therapy, functional genomics, contributing to understanding of genetics and development of new medical treatments. So, this completes mammalian artificial chromosomes. So, quickly wrapping up, we talked about artificial chromosome, we brushed up the little concept of artificial chromosome, we talked about the types of artificial chromosome, then we started mammalian artificial chromosomes. There we first studied about what are the elements that are present in artificial chromosomes so that they can mimic the natural human chromosome. The history of mammalian artificial chromosome, how the first human artificial chromosome was constructed. Then we talked about the steps that are associated to construct a human artificial chromosomes. Then we talked about the methods, various methods that are used for the construction of MACs, and finally we talked about the various application in details which can be there for the mammalian artificial chromosomes. Thank you very much. We will meet again in the next lecture.